The forgotten vitamin that rebuilds brain cells after 60. There is a vitamin that has been studied for decades. Hundreds of peer-reviewed papers document its relationship to brain cell survival, myelin integrity, and neurological function with extraordinary consistency. And yet the majority of adults over 60 have never been told about it in any meaningful clinical context. Not because the science is weak, the science is remarkably strong, but because this vitamin does not generate the headlines of trending supplements or pharmaceutical drugs. It is quiet. It is old, and it is being forgotten at precisely the moment the aging population needs it most. Here's the number I want you to hold in your mind through everything we cover today: 20 to 40%. That is the estimated proportion of adults over 60 who are deficient in this vitamin at levels that directly impair neurological function, not borderline low, deficient in ways that produce measurable physical changes in brain tissue, visible on MRI scans as accelerated brain volume loss and hippocampal shrinkage. That vitamin is B12, and today I am going to show you what it does inside your brain at the cellular and anatomical level, with a precision that permanently changes how you understand your own cognitive health. In our previous videos, we covered the glymphatic system and overnight brain repair, cerebroscapular blood flow, and the 5 foods that restore it, and the 2 cellular nutrients, choline and CoQ10, that power memory at the synaptic and mitochondrial level. Today, we go into the methionine cycle, myelin synthesis, and DNA repair inside your neurons, and identify exactly why B12 is the forgotten foundation holding all of it together. Introduction. Vitamin B12 is classified as a water-soluble B-complex vitamin, but that classification undersells what it actually is biochemically. B12 is a coenzyme, an essential molecular cofactor for specific enzymatic reactions that cannot proceed without it. 2 of those reactions are so fundamental to the structural integrity of your neurons that their failure, in the absence of adequate B12, produces consequences ranging from subtle cognitive slowing at the mild end to irreversible neurological damage at the severe end. We are going to follow B12 from the moment it enters your body to the moment it reaches the neuron and does its work. The problem: what B12 deficiency does to the structure of your brain. The brain changes associated with B12 deficiency are not abstract biochemical shifts. They are structural, anatomical, and progressive. The most documented neurological consequence of B12 deficiency is subacute combined degeneration, a condition in which the myelin sheaths surrounding nerve fibers in the spinal cord and brain begin to break down. Myelin is the fatty insulating layer wrapped around axons that allows electrical signals to travel at high speed between neurons. It is not a passive coating. It is a metabolically active structure requiring continuous synthesis throughout your lifetime, and B12 is an essential cofactor for the enzyme that drives that synthesis. Specifically, B12 is required for the function of methionine synthase, an enzyme that converts homocysteine back into methionine. Methionine is then converted into S-adenosyl methionine, SAM, the body's universal methyl donor. SAM donates methyl groups to the phospholipids and proteins that form myelin. Without adequate B12, methionine synthase slows, SAM production falls, myelin methylation is impaired, and the insulating sheath around your nerve fibers begins to thin and fragment. You do not need to reach the severity of subacute combined degeneration to experience real brain consequences from B12 insufficiency. The same myelin synthesis impairment that produces dramatic neurological damage at severe deficiency produces subtler but measurable changes at the moderate insufficiency levels, affecting 20 to 40% of adults over 60. A study published in the journal Neurology used MRI imaging to compare adults with low B12 levels against those with adequate levels over 5 years. The low B12 group showed significantly greater brain volume loss, with the greatest shrinkage in the hippocampus and cerebral cortex, the 2 structures most responsible for memory formation and higher cognitive function. The Oxford University study followed adults over 70 with mild cognitive impairment for 2 years. Half received high-dose B vitamin supplementation, including B12. Half received placebo. The B vitamin group experienced 30% less brain atrophy than the placebo group, 30% less physical brain shrinkage from a vitamin. That finding remains one of the most significant and most under-discussed in all of nutritional neuroscience. You have been told that brain shrinkage after 60 is simply what aging does. The truth is that for a significant proportion of older adults, a measurable and correctable nutritional deficiency is accelerating that process, far beyond what aging alone would produce. The hidden truth: homocysteine, the toxic molecule destroying your neurons without B12. Inside every neuron in your brain, the methionine cycle operates continuously. Methionine is converted into SAM, which donates its methyl group to critical biological reactions and becomes homocysteine in the process. Homocysteine is a temporary intermediate that should be rapidly recycled back into methionine through methionine synthase. That enzyme requires B12 as its essential cofactor. Without adequate B12, methionine synthase cannot function efficiently. Homocysteine is not recycled. It accumulates in the cell and spills into the bloodstream. Elevated homocysteine, hyperhomocystinemia, is one of the most directly neurotoxic biochemical states the aging brain can be in. Homocysteine damages neurons through three simultaneous mechanisms. The first is excitotoxicity. Homocysteine binds to and chronically overactivates NMDA receptors, the glutamate receptors on postsynaptic neurons, involved in long-term potentiation and memory formation. Chronic NMDA overactivation allows excessive calcium to flood into the neuron through its associated ion channels. That calcium overload activates destructive intracellular enzymes, proteases that break down structural proteins, lipases that degrade cell membranes, and endonucleases that fragment DNA. The neuron undergoes excitotoxic death. The second mechanism is DNA repair impairment. Neurons are postmitotic cells. They do not divide and replace themselves. They depend entirely on DNA repair mechanisms to maintain genomic integrity across a lifetime. Homocysteine inhibits the enzymes involved in base excision repair, one of the primary DNA repair pathways in neurons. When DNA damage accumulates faster than it can be repaired, neuronal function deteriorates irreversibly. The third mechanism is cerebroscapular damage. Homocysteine promotes oxidative stress in the endothelial cells lining your cerebral blood vessels, impairs nitric oxide production through EENOS, and accelerates atherosclerotic change in cerebral artery walls, compounding the direct neuronal damage with simultaneous vascular deterioration. A meta-analysis of multiple large population studies found that for every 5 micromoles per liter increase in plasma homocysteine above normal, the risk of Alzheimer's disease increases by approximately 35%. This is a dose-dependent relationship between a measurable, correctable biochemical marker and the risk of the most feared form of cognitive decline. And the most reliable intervention for lowering homocysteine is restoring adequate vitamin B12 status. The solution: natural food sources of B12 and the absorption crisis after 60. Before discussing food sources, I need to explain the absorption anatomy of B12, because this is what makes deficiency so specifically prevalent in adults over 60, even in those eating apparently adequate diets. B12 absorption requires a precise, multi-step sequence. In the stomach, gastric acid and the enzyme pepsin liberate B12 from food protein. The freed B12 binds to a carrier protein called haptocorin. In the small intestine, pancreatic enzymes digest the haptocorin and release B12 again. B12 then binds to a glycoprotein called intrinsic factor, produced by parietal cells in the stomach lining. The B12 intrinsic factor complex travels to the terminal ileum, where cubolin receptors absorb it into the bloodstream. After 60, this entire sequence fails at multiple points simultaneously. Gastric acid production declines through a condition called hypochlorhydria, impairing pepsin activity and B12 liberation from food protein. Studies estimate that up to 30% of adults over 50 have sufficient hypochlorhydria to meaningfully impair food-bound B12 absorption. Simultaneously, parietal cell function declines, reducing intrinsic factor production. Without intrinsic factor, even liberated B12 cannot be absorbed in the terminal ileum. Many older adults also take proton pump inhibitors for acid reflux, which further suppress the gastric acid that initiates the entire cascade. The result is an adult over 60 eating what appears to be an adequate diet who is nevertheless developing significant B12 deficiency, not from insufficient intake, but from a gastrointestinal system that has lost the capacity to extract and absorb it. The single richest dietary source of B12 is beef liver. 3 ounces of cooked beef liver contains approximately 70 micrograms, nearly 3,000% of the daily recommended intake. The concentration is so extraordinary that even with significantly impaired absorption, enough gets through to make a meaningful biological difference. Consuming beef liver once or twice per week is one of the most powerful single dietary interventions available for B12 repletion after 60. Shellfish are the second exceptional category. 3 ounces of cooked clams provides approximately 84 micrograms of B12. Oysters provide approximately 32 micrograms per 3-ounce serving. Mussels provide approximately 20 micrograms. These concentrations make shellfish among the most efficient B12 delivery vehicles available through whole food. Fatty fish are the third major source, and the overlap with our previous videos is clinically significant. Salmon provides approximately 4.8 micrograms per 3-ounce serving, alongside DHA for endothelial membrane health, choline for acetylcholine synthesis, and CoQ10 for mitochondrial energy production. Sardines provide approximately 7.6 micrograms per serving. Mackerel provides approximately 16 micrograms. Absorption from fatty fish is generally better preserved in adults with mild hypochlorhydria than absorption from red meat. Eggs provide approximately 0.6 micrograms per whole egg. The B12 in eggs requires less aggressive gastric acid liberation than meat-bound B12, making eggs a consistently absorbable daily source for adults with reduced stomach acid. 2 whole eggs daily make a reliable ongoing contribution to B12 status. Dairy, specifically milk and plain yogurt, provides approximately 1 microgram per cup. The B12 in dairy exists in a free or loosely bound form that requires minimal gastric processing for absorption. For adults with known hypochlorhydria, dairy represents the most reliably absorbed daily B12 source and should be prioritized alongside higher concentration sources. 2 strategies meaningfully improve B12 uptake in older adults. First, distribute B12-rich foods across daily meals rather than concentrating intake in one large weekly serving. B12 absorption through the intrinsic factor pathway is saturable at approximately 1.5 to 2 micrograms per meal. Daily distribution is significantly more effective than infrequent high-dose consumption. Second, for adults with significant hypochlorhydria, sublingual methylcobalamin bypasses the entire gastric absorption sequence. B12 absorbed under the tongue passes directly through the mucosal capillaries into the bloodstream without requiring stomach acid, intrinsic factor, or cubalin receptors. What to avoid? 3 things actively blocking your B12 and accelerating brain cell loss. 3 common factors are actively depleting or blocking B12 in adults over 60 at the precise stage of life when adequate B12 is most critical. The first is long-term proton pump inhibitor use. Proton pump inhibitors irreversibly block the hydrogen-potassium ATPase enzyme in the parietal cells of the stomach lining, eliminating gastric acid production, and with it, the first essential step of B12 liberation from food protein. Studies consistently show that 2 or more years of proton pump inhibitor use significantly increases the risk of B12 deficiency. If you have been on a proton pump inhibitor for an extended period, ask your physician specifically about B12 monitoring. The second is a strictly plant-based diet without deliberate supplementation. B12 exists only in animal-derived foods, produced by bacteria and accumulated in animal tissue through the food chain. No plant food provides biologically active B12 in amounts sufficient for neurological protection. Adults over 60 on a vegan diet who are not supplementing B12 consistently are at high risk of the exact brain changes discussed throughout this video. Sublingual methylcobalamin is non-negotiable for brain protection in this population. The third is chronic alcohol consumption. Alcohol damages the gastric mucosa over time, impairing the parietal cells that produce stomach acid and intrinsic factor. It impairs absorptive function in the terminal ileum, and it disrupts B12 utilization at the cellular level by interfering with the methionine cycle directly. Chronic alcohol consumption damages B12 status through 3 simultaneous mechanisms: reduced liberation, reduced absorption, and impaired cellular utilization. Final teaching. Let me bring every thread of today's teaching into one clear anatomical picture. Vitamin B12 is the essential cofactor for methionine synthase, the enzyme driving the methionine cycle, producing SAM, supporting myelin synthesis, and preventing homocysteine accumulation in your neurons. When B12 is insufficient, myelin breaks down around axons, reducing signal transmission speed. Homocysteine overactivates NMDA receptors, triggering excitotoxic neuronal death. DNA repair in postmitotic neurons is impaired. Cerebral endothelium is damaged, and brain volume loss accelerates at a rate the Oxford study showed can be reduced by 30% with adequate B12 restoration. After 60, absorbing B12 from food becomes progressively more difficult as gastric acid declines, intrinsic factor output falls, and commonly prescribed medications further impair the absorption cascade. The solution requires choosing the highest concentration of natural food sources: beef liver, clams, oysters, fatty fish, eggs, and dairy, distributing them across daily meals, and for those with significant absorption impairment, using sublingual methylcobalamin to bypass the failing gastrointestinal system entirely. Your action point this week is specific. Add one B12-rich food every single day. Begin tomorrow with 2 whole eggs. Include salmon or sardines at lunch or dinner. Add plain yogurt daily. Include beef liver or shellfish at least twice this week. That combination across 7 days will deliver more B12 to your neurons than most adults over 60 receive in an entire month of typical eating. Ask your doctor this week for 2 specific tests: a serum B12 level and a plasma homocysteine level. Serum B12 alone can miss functional deficiency in adults whose B12 appears adequate in the blood, but is not being utilized efficiently at the cellular level. Homocysteine tells you the functional story, because elevated homocysteine confirms that methionine synthase is not working adequately, regardless of what the serum B12 number shows. Both tests together give you the complete picture of your B12 status at the level that actually determines your neurological health. Tell me in the comments, have you ever had your homocysteine tested? Do you know your number? That number may be the single most important piece of information you can have about the current biochemical state of your brain. If you have never been tested, this video is your reason to change that this week. In our next video, we are going inside the gut-brain axis, the direct anatomical highway between your digestive system and your hippocampus. The enteric nervous system, the vagus nerve, the microbial production of neurotransmitters, determining the neurochemical environment of your memory center right now. The connection between your gut microbiome and your cognitive health is more direct and more actionable than most people have ever been shown. Subscribe so you do not miss it. Share this with someone over 60 whose brain is waiting for this information. And remember, your neurons are not simply deteriorating because of time. They are deteriorating because of a deficiency that has a name, a mechanism, and a solution. Now you know all 3.